We're good? Okay. Good afternoon and welcome to the Science Electron Microscopy Center's Winter EM course. We are now moving beyond our standard basics and three modalities into EM challenges and EM frontiers. And this is one of the most exciting aspects of the course because we're not talking about what has been done already, we're talking about how EM is being extended for the future, and we're moving forward from there. One of the aspects that you heard from yesterday is validation, and we're going to hear more about that from our lecturer today, Kathy Lawson from Rutgers, in the Department of Chemistry and Chemical Biology, I believe. Is that true?
Used to be.
Used to be. Is it now? What is that?
It's Institute for Quantitative Biomedicine.
Quantitative Biomedicine. And it is. Is it? So the main thing that Kathy is really passionate about and has been working on are databases, and you would think, how useful is that? Very useful. You all maybe have used the PDB, the EMDB. There's now integrated into a one-dep system, and that is how you make a resource for scientists to really push biology forward, because all the data that we've been accumulating, everything that we've been depositing, is now trying to be unified and being made more accessible. And that is a real trick. Would be, as a researcher, you read a paper or you find an interesting piece of research and you want to go back to the primary data. What is that primary data? What should be archived? And in what format is a format that you can make use of? And we'll hear a lot about that today, and also about the growing need for standards in our field. And that is something that Kathy has really been leading the charge for, and with others, and trying to develop for the EM space as we're moving to a different resolution regime. How do we match what other modes of structural biology have been using, and how interchangeable, and how can we interface with that? So our speaker today is Kathy Lawson.
Thank you so much, Ed. It's a pleasure to be invited back to speak in this course. I actually have finally learned how to get on the A train and get up here without getting lost, which is a great achievement for me. Okay, so I'm going to tell you about the work I've done under the umbrella of the Unified Data Resource for 3D-EM project. I'm told I need to use this. Which we have actually recently renamed from EMDataBank to EMDataResource. Just the slight little switch, because EMDataBank means to most people the data archive for EM maps, and we realized that that was kind of problematic because really our purview has been to develop data archives for both EMDB and PDB. And so, so we made the decision to call it EMDataResource instead of EMDataArchive, but it's been an ongoing project since 2007. Our fearless leader is Hua Chu. He was, of course, at Baylor in Houston, and I really enjoy visiting him in Stanford a lot more now that he's moved. And we also collaborate very heavily with the group at EBI. All right, so what are the data archives that are available for 3DM? And so hopefully you are familiar, but if you're not, so the one that you know, sort of the longest going archive that is available for fitted models is Protein Data Bank, of course. And in around 2002, the EBI developed this EMDataBank archive just for 3DM volumes. And more recently, really, I'm just thinking maybe 4 years ago or so, EBI also developed this Empire database, and it's for raw images. And somebody here was asking me this question just a few minutes ago, just about, well, what is the worth of depositing raw images? Well, it may be useful to you, or it may be that it's really useful for the people in the field You need to have benchmark data to be able to develop better programs, better algorithms. So you always need to have some reliable source of data. You don't necessarily need to deposit raw images for every single structure that you determine, but it's useful to have that available. And if you know, people express particular interest in having your raw images, you may want to consider depositing to Empire. Now, if you want to publish your work now from a 3DM experiment, you will need to deposit your map to EMDB, and if you have a coordinate model, it will need to go to PDB. Most journals do have those requirements now. So I've actually been involved in this project since 2007, which was, you can see, there was not very much, and all the structures were kind of blobby at that point. It was very exciting to see the rapid growth in both the EMDB and EM structures in the PDB. And equally exciting was when we decided we wanted to publish about our work, and in 2011 we had an initial publication about EMDataBank. In 2006, a second one, and we had such a great winning cover scheme that we decided to go for it again, but you can see such a huge difference between the two. So you're going from basically blobs to you know, structures where you can really see the details. And this is actually available on our website under the statistics page every week. It is now updated. So like 2009 is just this is just partial data at this point because it's only through April. But you can see how the resolution range has changed over the years. So at the beginning, worse than 20 was really what we were getting, and then all of a sudden the revolution has come, and unbelievable, more than 500 maps in the 3 to 4 angstrom resolution range last year. Well, you probably have seen it in the literature, right? It's just an explosion of huge interest in using this technique for really exciting and useful biology, structural biology. So how do you find a cryo-EM structure that you might be interested in looking at? So each data archive service, usually one of the important things that they do is try to make the data findable. So we list here on our website, on the search page, all the different possibilities. And maybe I'll just use this. And so we have a search here at our website. We've actually just updated our search. We had a search that had been installed in 2007, which worked really well with those few tiny, small number of structures that were available. And it's, you know, so over 10 years, the software got severely outdated and the machine can no longer be supported. So we made a totally new search and made it available just a couple months ago. So I'm going to tell you a little bit more about that. But you can also search at other locations, and you can search for models instead of maps. These two services allow you to search for both maps and models at the same time. Again, if you're interested in raw image data, go to Empire. So our new search is not really geared towards, let's say, beautiful looking results, but what we really aim to do is to try to give you access to as much useful data as possible in the form of tables. And so you should be able to, if you go to this page, you should be able to enter an ID, either EMDB or PDB, an author name, a species name, or a topic. And you can also filter your results. So the default is released entries, but you can also search for entries that are on hold or that are obsoleted. You can also filter by whether it's single particle or helical or tomography, and what kind of specimen, whether it's, again, a single, a particle or an array or a helical array. And you can also specify what resolution range you want to look at. And you can pick various options for what you want in terms of results. And in addition, so I tried to make it so that the box itself, and when I say I actually designed it, so and coded it. So the box itself it has an autocomplete and should hopefully help you to find whatever you're searching for, but we actually do also have keyword options. So you can actually go in and really dig into different kinds of searches. And there's this funny thing about ranges with if you have just the square brackets, it means less than or equal than or greater than or equal to. And then if you have a curly bracket, it means just less than or greater than. So it's a little fine detail. But anyway, I encourage you to test it out, and should you feel that there's something that you really would like to search for that you can't, we'd be very happy to hear from you. And we'll try to implement something that will help you out. All right, so another facet that I've been very involved with since 2007 has to do with deposition. So in 2007 we had, there was a capability to deposit maps in EMDB, and there was a capability to deposit models in PDB. And we had this kind of historic meeting where Helen Sable got up and said, I need a one-stop shop. I need to be able to do both at the same time. And so we implemented an early version of that where people could deposit their maps, and then they could push a button, and the metadata that was entered for microscope, all your imaging, all of your reconstruction information would get transferred over to the PDB and get included with that metadata as well as what was in your map. Since 2016, things have been even better. We've retired that initial joint deposition system. It worked really well for 10 years, but we knew it was temporary and we wanted to have a more permanent solution. So the worldwide PDB, our group worked with Worldwide PDB to develop a joint system that handles X-ray, NMR, and EM methods. And this has been available since 2016. And it's no longer the serial deposit your map and deposit your model, but it's truly joint in that you set up a deposition and you're actually assigned both an EMDB ID and a PDB ID with the same session. and a validation report is produced. It's a little bit minimal right now, but we're working on improving that. So all right, so when you come to this deposit.wwptv.org, what you get initially is you're asked to define your experiment. So for electron microscopy, you would pick one of these options, and then it goes and asks you these key questions. Are you depositing coordinates? Has the associated map been deposited previously? And depending on what you answer, you get, your requested accession codes are automatically assigned for that session. You don't actually get the codes until you complete your deposition. But one thing that we had to prepare for, so EMDB IDs were EMD-4-digit numbers, and we're running out of 4-digit numbers. So the last time I gave this talk, I said coming soon, but I'm sorry, it's here. It's already here. So if you see 5 digits, don't get confused. We're keeping the 4-digit IDs for those that fit into 4 digits, and we're just going to expand as necessary. So yeah, someday we'll have as many entries as PDB, which has 150,000 now. So when you're making a 3DM deposition to the one-dep system, EM depositions are a little bit more complex than X-ray or NMR because, well, in part it's because EMDB actually allows you to deposit a number of different kinds of files. And so, of course, coordinates are going to end up in your PDB entry. You have to give a map. And I often get asked the question, well, what map should this be? That your main map, the one that is primarily associated with your EMDB entry, and the answer I usually give is it's the map that you have interpreted to give you your biological inferences or your coordinates. EMDB you're going to be able to make a much nicer picture of your map than we can. We can put it up on Chimera and take a snapshot of it, but it will be kind of bland looking. So we ask that people provide an image along with their map. It makes for an interesting, so every week with our website, we put all the new releases out on a little carousel, so you can see the images as they come by. So give us nice images. Additional maps. Now those can be, for instance, the raw map that came out of your reconstruction program before you apply, let's say, B factor correction or masking or some other correction that helps you to do the interpretation. And I'll mention here already another. Oh, let's see. No, I'm sorry. We already have another category here for half maps. So we already asked for those separately. You can supply masks, and if you used a mask to help you interpret the map, then it would be really useful to, in general, for you to have that up in case somebody wants to also apply the mask. Half maps, structure factors, and That can be because, that can actually be either because you did a diffraction experiment and you actually have 2D or 3D crystal structure factors, or we do also accept structure factors that were computed from maps. And finally, we accept this FSC file. What is that? It's just an XY points curve. And then finally, if you have the ligand, an image of your ligand is usually useful to help the PDB figure out how it should be represented chemically. So for the FSC curve, there are a number of different options. There's an XML format that it needs to be supplied in, but software packages in general will produce that format for you. Or you can use PDDES server to produce the FSC. This plot actually came from their server, and so it's just an example. So it gives you the curve and also a number of different options for interpreting it. And then, well, this is sort of getting ahead of myself a little bit, but what I keep hearing from the community now is uploading the half maps is really important, and is probably going to be, there's likely going to be a recommendation that is mandatory. So, at least for single particle work. All right, then just a little note. We have EMDB actually releases files, or let's say the metadata of the data that describe the experiment is the file is in XML format, and while the PDB produces this PDBX MMSIF format file, and I just want to make sure everybody doesn't have the concern. We make it a point to make sure the underlying dictionaries are equivalent. It's a little bit more work for us, but why do we have different format files? It's sort of a historical thing. But here's an example of the dictionary for vitrification instruments. Exactly the same lists exists for both the XML or the XSL schema against which the XML files have to validate, and the MMSIF dictionary against which the MMSIF files have to validate. And we did make a point of adding Spotaton, as we know.
Well, about Chameleon, that's the commercialized version.
Oh, okay, we better add it. Is it, they've sold a number of them now, or do you know?
They sold half a dozen that early production models. I don't know when they're actually heading out. Maybe this year, sometime this year.
Okay, all right, cool. All right. So then lastly, what we've been involved in this project is trying to, I guess, sort of engage the community in discussions about validation, because, you know, it's a much younger field than, for instance, NMR or X-ray crystallography. But it's becoming a very prominent field, and so you would like for people to believe your structure if you publish it, right? So how can that happen? So in 2010, we had sort of the first meeting of this type, where we invited people to come to Rutgers to tell us what they felt was the current state of the art in terms of validation. And here we produced, in 2012, there was a white paper that came out. And I would say, so here I've just summarized the two main recommendations. One, the first was that the model stereochemistry should really be analyzed the same way as it's done for X-ray and NMR, and that of course makes sense because, you know, an amino acid side chain is an amino acid side chain, no matter how you're imaging it or measuring distances. And I still recall sitting in the room and there was a lot of heated discussion about how to validate maps at the time. So this ended up really being the recommendation. The main recommendation was that more research was needed. But actually the sort of the gem of the idea for this whole idea of, some people call it a gold standard and some people say no, you shouldn't call it that, but you know, independent half maps that you hopefully you guys have been exposed to that in prior classes, right? Yeah. So the idea is that, you know, you completely separate the, you know, your particles into two bins, and you produce two completely independent maps. And you know, so if you to the extent those are similar to each other, sort of gives you the extent to which you can really trust your result. So the current validation report that we instituted as a result of that meeting actually is pretty similar. It's pretty simple. So we give the resolution value that's reported by the depositor. So at this point, we're just trusting them to tell us what the resolution is. And we produce a simple set of model statistics. There's this nice slider graphic that gets produced. So this particular structure had no side chain outliers. It was Ramachandran outliers is kind of an interesting one because ever since that was instituted fairly early on for X-ray, and everybody who does X-ray structures makes sure they have no Ramachandran outliers unless they can possibly help it. So here, 0.5% is a very reasonable result actually, but it gets slid all the way over here because there's so many structures that have it. Yeah, so, and class score, that's actually not such a bad class score either for this particular structure. But we compare both to all structures and to all EM structures. So we wanted to go further with this, and we decided to take an approach which should engage the community in a set of challenges. So we sponsored 2 challenges in 2015 and 2016, a map challenge and a model challenge. And the goals were, I guess, number 1, to develop benchmarks for study so that, you know, different groups could sort of compare the same data and decide what kind of metrics make sense to analyze the data. we wanted to encourage development of best practices, evolve criteria for validation, and to compare and contrast different approaches. And we wanted to make use of recent data that was archived. If you look at these structures, you know, these were some of the early structures that came out of the resolution revolution that were very exciting at the time. In hindsight, we chose too many targets, number one, and number two, it was very hard to manage two challenges at the same time, and it ended up taking a lot more time than we hoped that it would. So we did fairly parallel development of the two challenges, and we had a set of people who we designated as the challengers, who supplied their best possible map using raw data target data, and we also had people provide their best possible models using in the challenge phase against maps that we assigned. And then we went through a process of trying to do some assessment of each of the results, and we ended up having a pretty big meeting in October 2017. I think it's shown to you here. These were all the people involved. So we're about, I think, 90 or so participants who showed. And then it took another some months, a year to produce, and we were able to produce a virtual special issue of the Journal of Structural Biology, where there are 18 peer-reviewed articles and editorial by myself and Wachu, summarizing outcomes of the workshop and recommended best practices. And did you have a question? Or? No? Oh, okay. So there's a lot in that journal special issue, so I would encourage you to actually go and take a look. But just to give you an idea, so the APO Ferritin Target turned out to be one of the most interesting in terms of results coming in from the challenge, because so this red line represents the Russo and Passmore results. So they were able to get around 4.6 angstrom resolution or so. Perhaps the reason that that particular one is interesting is because, or the reason that they didn't push it was because they were it was done with these gold grids, and they didn't do any movie frame alignment, and they just want to say, hey, look how good we can get just by doing the, changing to gold grids. But then it turns out that if you do the gold grids and you add the movie alignment, you can do considerably better. And so quite a few of our challengers were able to do better than the sort of the benchmark study. And I have on the next slide, and this just highlights one of the issues that we currently have. And so I mentioned to you that we asked the depositors to tell us what their resolution is, right? So it's basically it's a depositor reported resolution. And so I would challenge you to tell me which one was reported at 3.1 versus 3.5. Yeah. So, and this was a key outcome of the challenge, really, is that different well, I mean, we already had the sense, but we could really kind of quantitate it now, that different expert practitioners can arrive at different resolution estimates depending on how they did their estimation. And so that leads to a question. So could the community agree on a standard for resolution estimation? And I'll talk about that. I have a bit of an answer for that. That'll come a little later. Some other results that we learned from the MAP challenge. The good news, all major packages that were represented, and there were like 15 or more of them, were able to produce maps of equivalent quality. Of course, some of them were, Relion was overrepresented. And there were some that were underrepresented, but really most major packages can basically produce maps that are good. But the difference in quality is really among the practitioners. So, I mean, there are particular people who are able to develop their own software and are able to, they know how to manipulate it, right? And so they're really able to produce maps that are quite good with their software. And the trick then is, how can they transfer that information to other people who want to use their software? So that is an issue. And as I mentioned, the reported resolution issue. And so we concluded the FSC use of FSC is inconsistent, and we really need to have bulletproof workflows and best practice standards. So some of the things you will read if you go to this special issue delve into this in more detail. Then I also want to mention about this workshop we had in January. So this was actually an initiative. It's sort of it's a parallel initiative to ours that was started just recently in the UK. It's led by Peter Rosenthal and includes people at EBI and at other institutions in the UK who are similarly with us are interested in developing validation standards for EM. So they held a workshop in January, and I was very encouraged by this because I guess hopefully there is a white paper forthcoming, and there was a lot of terrific discussion, a lot of consensus. But a key recommendation that came out of this was that really the EMDB should be producing some kind of a resolution estimate based on deposited half maps. So I'm very excited and hopeful that that will actually come to pass sooner rather than later, because I think that will really help the whole issue of resolution not being a completely reliable metric. So then I have a summary here of some EM structure validation servers, both for maps and for models. If you're interested, I don't know if you're familiar with these, but and maybe Tom Waltz might have talked about the tilt bear one, because that's really for low.
He did talk about it.
Yeah, right. So that's really for a low resolution. So if you just if you're not really sure about the overall shape or hand of your structure, then it can be useful to use this tilt pair server. Of course, that means you have to produce the tilt pairs. That's a separate issue. Then there are quite a few things that have popped up in terms of local resolution, and maybe that could be an interesting discussion point for the students. Further, I actually have noticed that just about every single structure that gets published these days has some kind of local resolution figure that goes along with it, and so that the idea is to, I guess, either prove that you're not over-interpreting your map, or to give caution about particular areas of your map, which may not really be as good as other parts. And if you have a domain that's, maybe not, kind of floppy with respect to the rest of the structure, like I think there's that stalk in the ribosome that tends to, just sort of spread out. Then it's very useful to have local resolution. I don't think that's something that is, we haven't really had a lot of discussions about whether that needs to go in the data archives or not.
I have a question.
Yes.
So right now, would you say that most people who are publishing, they report their best resolution?
It's true, yes.
So then in your opinion, do you think it makes sense to be reporting an average resolution?
But I guess, what would you mean by that?
So like, I think if you, usually people will hopefully be reporting a resolution that is correspondent to the biological question that they're trying to answer. So if they have a 3 angstrom resolution at the core, but then they're trying to answer questions at some periphery, like, does it really, like, should there be some sort of standardization to, like, kind of meet in the middle? Like, you don't have to report like that 8 angstrom movement or whatever, right? But like something that might feel a little bit more transparent than like reporting some super high number.
Right. Yeah. I mean, I think if you're doing an FSE analysis, I guess it may be weighted more towards sort of the better part of the structure, but still that piece that is not
totally resolved,
not totally resolved is not going to, should in principle contribute. So I think it's not a
big issue.
I'm not sure. Other people might have another opinion, but this Scipion local resolution is interesting because it has, I think, 3 different ways to calculate your local resolution. For models, we have, you can actually go to the WPTV and upload a structure, and it will produce a validation report that's exactly like the one that you would get if you went to PDB. You may be familiar with Molprobity as well. That can be used to analyze the quality of your model. EM Ringer is one interesting sort of newer method that you might be able to use and it's actually an interesting, what is it really validating is kind of an interesting question, because it's really kind of validating both the map and the model at the same time. I like to think of it as sort of a backbone bumpiness measurement. So sort of the more, sort of the higher resolution of a protein structure that you would have, the more you will see, you know, you won't see a smooth tube, you'll actually see, you know, see beta bumps coming out, and it gives an interesting perspective on that. And, you know, there are, with malprobity, there's actually this new, Jane Richardson has been promoting this new metric called KBLAM, and that may also be useful. So one of the things that we're doing this coming up, and I don't actually have a slide on it, but we are actually moving forward with a sort of a new round of model challenge, in which we've decided we're really paring back just in terms of the number of people involved, the number of targets, but we're gonna actually have people sit down and focus on just a few models, 2 targets, and does this metric make sense at this resolution? Does this metric make sense at this resolution? And hopefully we'll come up with some recommendations following that. And I think this is the group of people that I'm currently working with. So Wah, again, at NCMI, which is now at Stanford, and working with Helen Berman at Rutgers, and with the EMDB group at EBI. And here's some references I would recommend in particular if you want to follow up. So this is the editorial for that special issue. And this is a recent review about databases and archiving for Cryo-EM that I wrote with Arden Patwardhan. And I think this is still relevant, this the Henderson et al. 2012. So with that, I'll just open it up for discussion. Oh, one more thing. I promised I would mention RCSB, PDB. My colleagues at Rutgers are hiring. So we're hoping to find an annotator and a front web end developer. I didn't actually say so much about what happens when you deposit your structure, but annotators, we have a professional staff of people that look at every single entry that comes in and make sure that all the metadata makes sense. We look for EM structures, we actually look at the map and the model and make sure that they are superimposed in the same position. And what else?
What is the minimum requirement to submit if, let's say, there's proprietary research or there's non-disclosure concerns? What is the stipulation on that? It's a public database, so you would want it freely available, but what happens if someone wants to deposit but has concerns?
Oh, well, there's no option really to hold the data infinitely long. You are allowed to hold data until either publication or for 1 year, whichever is shorter. So, yeah, so if there's proprietary concerns, then I think, well, like, yeah, then you, it doesn't go in here.
Yeah.
But you know, maybe eventually it would, you know, after the proprietary concern is passed, and say, you know, you were working on some project to develop you know, on a target, on a drug target, a hot drug target, and maybe 10 years later or 5 years later, you know, that project is over, and you could, you know, so you could deposit it then. Yeah, PDB and EMDB, you know, we don't, yeah, it's all public, so. Good question.
So for
Yep.
the validation servers that you showed, so you upload your half maps and then those will do some sort of resolution determination?
Yeah.
How do they validate exactly?
PDDES FSC server, you upload your half maps, and I think you have to provide just like a little bit of additional information, like what percentage of the map your structure actually fits in, and maybe symmetry. But then it produces that curve for you. And these other servers, I think most of them also work off the half map. I'm sorry. ResMap, just I think is, you only need to provide one map. And they do some kind of a statistical analysis on the, I guess sort of the per pixel variation to get an idea of whether, of what the resolution is at a particular location.
Is there more than one resolution determination method that they use, that there would be multiple opinions on a resolution of one particular map?
So I think the biggest issue really has to do with I guess sort of the parameters that get used for the determination. And one interesting factor is just sort of the box size of that you're using for the particle. So if you have a large box size versus a smaller box size, that alone can change your FSC result. So I don't know if in let's say here, do you have recommendations that you give people for what box size they should use for their particles? Is there kind of a standard for that?
There's a rule of thumb, like 1.5 to 2 times your particle size.
Yeah.
But then you might also have a mask. So
Yeah, and so if people mask, then that also will change the outcome. So I think the recommendation is actually going to be, it's not going to be no masking, but sort of minimally masked. So I know a lot of programs actually do some kind of a soft spherical mask, so that even the map that comes out from the reconstruction software already has that. So it's like you don't really even have a choice. It's already applied.
With CryoStorm, we'll iteratively create one that ends up resembling the shape, the overall shape of your particle pretty closely.
Oh yeah. And that could be, that could be very useful for you for doing your, doing your interpretation. But
do you know if that mask that it generates that's used for FSC calculations?
Yeah, they have different, one for the FSC and one for the final one. So one for every type one.
Yeah.
And you can upload them onto the server so they can check it. Yeah.
So the half maps that come out of your refinement jobs?
Yeah, non-mask. Yeah.
They're non-masked.
Mm-hmm.
But it'll report an FSC, a resolution estimate, based off of some final really tight masking that a lot of people will probably use.
Yeah.
But they say what their resolution is.
We upload that one because that's the best.
Yeah.
Tightest mask.
Right. Yeah. So the idea is that EMDB will generate this sort of metric that should, in principle, I guess it will depend what people upload for their half maps, but the idea is that you will be able to better compare between different entries. You can still provide your best resolution with your tight mask, and that will be a different, that'll be a different number. Yeah, that's kind of where we are. And I think in terms of map model fit, the interesting question is what kind of metric we're going to end up using for, say, a side chain. How do you assess the quality of fit of a side chain into side chain density? For X-ray crystallography, there's these, they have made, they have decided on their standards that they use. But it'll need to be different for EM because you produce your structures and you produce, the data is different, so it needs to be done differently. And decision, so it just needs to be looked at carefully and by experts and to decide what really makes sense. Any other questions?
So as you're getting more and more recommendations, you're seeing more and more, how do you keep on expanding the interface, and how much more data are you trying to keep? Because there's more validation standards, or there's more recommendations on data that you should also be annotating your deposition with.
Right, yeah, so, yeah, I mean, in terms of accepting more types of data, it will mean that the dictionary that supports the archive will need to be updated to accept the new values that will come in, and hopefully it can be done in a way so that a software program that produces that data can actually be imported so that that would actually be the best of all. So we have a software called PDB Extract. It doesn't really do much for EM right now, but for crystallography programs and NMR, it will actually automatically read the log files and pull all of the validation-related information into MMSIF for you, so you don't even have to deal with it. So I think in terms of requirements for storing, those kinds of things are pretty small compared to the maps are still some. It's interesting. We actually have, I'm trying to remember, I think the biggest map we have is like 40 gigabytes or something crazy thing like that. And so one of my colleagues at EBI was saying to me, should we impose a limit?
Yes.
Because actually for a tomographic map could also go in Empire, and Empire is really much better suited to take large amount, data that actually requires a lot of storage space. EMDB can be, well, I mean, it's growing very rapidly right now, but yeah.
Empire will take anything you want to give it. Like if you want to give it like a particle image stack instead of your all of your like raw micrographs. You do that.
So I think that the one restriction for Empire is that it has to be related to an entry in EMDB. But you can give it particle stacks and raw images and If you're doing tomography, you can give it the tilt series, and you can also give it the final map. All right.
Thank you.
Journal club next week? Or this Wednesday?
Yeah, it'll be practical this Wednesday, and then it will be on all these validation stuff that we're doing and me actually using some